Hello dear students, welcome back to my YouTube channel. Today we are going to discuss the topic restriction enzyme and how to isolate the DNA after cutting, okay, from the chapter biotechnology, principle and processes. है ना? So let's begin. Restriction enzyme, ठीक है? तो चलिए restriction enzyme क्या होता है? आप जानते हो कि जब भी आप किसी भी डीएनए को कट करने वाले हो तो उसके लिए आप न्यूक्लियस एंजाइम यूज करोगे क्योंकि न्यूक्लिक एसिड को कट करने वाला न्यूक्लियस होता है। अब न्यूक्लियस दो तरीके का हो सकता है, या तो वो न्यूक्लिक एसिड को बाहर से कट करे तो वो एक्सोन्यूक्लियस हो सकता है, या फिर सिर्फ अंदर से कट करे तो वो एंडोन्यूक्लियस हो सकता है। अब अंदर से कट करने वाला एंडोन्यूक्लियस का भी दो टाइप है। एक ऐसा एंडोन्यूक्लियस जो कहीं से भी रैंडमली अंदर में ही कहीं भी रैंडमली किसी पोजीशन पे अंदर में कट कर दे, सो दैट वन इज अ टाइप वन एंडोन्यूक्लियस। इसको हम रेस्ट्रिक्शन एंडोन्यूक्लियस नहीं कहेंगे क्योंकि रेस्ट्रिक्शन एंडोन्यूक्लियस का मतलब होता है कि कहीं भी रैंडमली कट ना करे, एक रेस्ट्रिक्टेड साइट पे कट करे। और आपको पता होगा कि ये जो हम लोग न्यूक्लियस पढ़ रहे हैं या एंडोन्यूक्लियस पढ़ रहे हैं, ये हमने डिस्कवर कहाँ से किया? बैक्टीरिया से किया। तो बैक्टीरिया के पास क्यों था डीएनए को कट करने वाला एंजाइम? क्योंकि उस पर वायरस अटैक करता है, अपना डीएनए जो है वो बैक्टीरिया के अंदर डाल देता है। तो बैक्टीरिया में इन्फेक्शन होता है क्योंकि अब वो वायरल डीएनए की कॉपीज प्रोड्यूस करने लगेगा अगर एक बार वायरस का डीएनए उसके डीएनए से इनकॉर्पोरेट हो गया तो बैक्टीरिया के पास एक डिफेंस मैकेनिज्म है कि जब वायरस उसके अंदर अपना डीएनए डाले तो बैक्टीरिया उसके डीएनए को काट दे। तो बैक्टीरिया उसके डीएनए को कैसे काटेगा? विद द हेल्प ऑफ द न्यूक्लियस एंजाइम्स। एक्सोन्यूक्लियस से वो उसको बाहर की तरफ से कट कर देगा, एंडोन्यूक्लियस से उसको अंदर की तरफ से कट करेगा। लेकिन एक प्रॉब्लम थी कि अगर बैक्टीरिया के पास डीएनए को कट करने वाला एंजाइम है तो वो बैक्टीरिया के खुद के डीएनए को भी कट कर सकता है। तो उससे बचने के लिए बैक्टीरिया के पास मिथाइलेज एंजाइम भी था। यानी कि अगर बैक्टीरिया अपने डीएनए में मिथाइल ग्रुप ऐड कर दे तो फिर उस पे न्यूक्लियस एंजाइम, एक्सोन्यूक्लियस एंजाइम, एंडोन्यूक्लियस एंजाइम एक्ट नहीं कर पाएगा। जिस भी डीएनए में मिथाइल ग्रुप ऐड हो गया है वो अब कट नहीं हो सकता है जो भी न्यूक्लियस है उसके द्वारा। ओके। तो बैक्टीरिया में पाया गया कि दो तरीके के एंडोन्यूक्लियस बैक्टीरिया में पाए गए। एक जो है टाइप वन एंडोन्यूक्लियस था जो कि कहीं भी रैंडमली कट कर दे रहा था, मतलब वो रेस्ट्रिक्शन एंडोन्यूक्लियस नहीं था। और उसके पास एक और खासियत थी कि वही एंजाइम, टाइप वन एंडोन्यूक्लियस एंजाइम ही बैक्टीरिया के खुद के डीएनए में मिथाइल ग्रुप ऐड कर दे रहा था। सो माय कंक्लूजन इज दैट द टाइप वन एंडोन्यूक्लियस जो बैक्टीरिया में था, दैट पर्टिकुलर एंजाइम एक्ट एज न्यूक्लियस एज वेल एज मिथाइलेज। बट जो टाइप टू एंडोन्यूक्लियस था, जो रेस्ट्रिक्शन एंडोन्यूक्लियस था, वो एक रेस्ट्रिक्टेड साइट पे ही कट कर रहा था, अंदर में कहीं भी रैंडमली कट नहीं कर रहा था। और वो मिथाइलेशन नहीं कर सकता है। द पर्टिकुलर टाइप टू एंडोन्यूक्लियस और द पर्टिकुलर रेस्ट्रिक्शन एंडोन्यूक्लियस डज नॉट एक्ट एज मिथाइलेज एंजाइम। ओके, फाइन। तो चलिए हम लोग इसको स्टार्ट करते हैं। इस चैप्टर में आपके क्लास 12 के बायोटेक्नोलॉजी में सिर्फ टाइप टू एंडोन्यूक्लियस, सिर्फ रेस्ट्रिक्शन एंडोन्यूक्लियस की बात की गई है, टाइप वन की बात नहीं की गई। ओके। सो इन द ईयर 1963 दो एंजाइम्स जो कि रिस्पॉन्सिबल थे किसी भी वायरस, किसी भी बैक्टीरियोफेज वायरस की ग्रोथ को रेस्ट्रिक्ट करने के लिए इन ई कोलाई बैक्टीरिया, उसको हमने आइसोलेट किया। एंड वन ऑफ दीज एडेड मिथाइल ग्रुप टू द डीएनए। ये क्यों यहाँ पे बात कर रहा है? क्योंकि दो एंजाइम जो है आइसोलेट किया गया, एक एंजाइम मिथाइल ग्रुप ऐड कर रहा था, दूसरा एंजाइम जो है वो कट कर रहा था। सो आप जानते हो कि जब भी मिथाइलेशन करने के लिए अलग से एंजाइम है, इसका मतलब है कि उस पर्टिकुलर बैक्टीरिया के पास कौन सा एंडोन्यूक्लियस है? टाइप टू एंडोन्यूक्लियस है, रेस्ट्रिक्शन एंडोन्यूक्लियस है। द लेटर वाज नोन एज द रेस्ट्रिक्शन एंडोन्यूक्लियस। लेटर मतलब किसकी बात कर रही है? जो कट कर रहा है डीएनए को, उसके नाम ले रहा है कि रेस्ट्रिक्शन एंडोन्यूक्लियस था। ओके। अब आप जानते हो कि किसी भी रेस्ट्रिक्शन एंडोन्यूक्लियस का नाम जिस भी बैक्टीरिया से उसको आइसोलेट किया गया है, उस बैक्टीरिया के जीनस और स्पेशीज के नेम के अकॉर्डिंग हम लोग उसका नाम देते हैं। और 1 2 3 4 जो भी उसके नाम में लगाते हैं हम रेस्ट्रिक्शन एंजाइम के नाम में, वो एक्चुअली उस लेबोरेटरी में वो कितना नंबर का एंजाइम है जो उस लेबोरेटरी से आइसोलेट हुआ, उस बेसिस पे करते हैं, राइट? तो देखिए, द फर्स्ट रेस्ट्रिक्शन एंडोन्यूक्लियर वाज हिंड टू। देखिए, फर्स्ट डिस्कवर्ड एंडोन्यूक्लियस वाज हिंड टू, राइट? बट वेट फॉर अ सेकंड. So as we were discussing that the first restriction endonuclease was hinted to because it was discovered first. That's why it's written that it was the first restriction endonuclease, but its name contains the number 2 because it was isolated later, like its isolation took 5 years, right? It was found that Hindu always cut the DNA at a particular point. That's why it's restriction endonuclease and not the simple type 1 endonuclease, right? So it always cuts at a particular point, that is at the palindromic sequence or at a restriction site, okay, by recognizing a specific sequence of 6 base pair. This specific base sequence was known as a recognition sequence for the Hint2. And beside the Hint2, today we know that there are more than 900 restriction enzymes which we have isolated from over 230 strains of bacteria. and each restriction enzyme has its own different recognition sequence, the particular palindrome which is recognizes to cut. But you don't have to remember all the 900 recognition sequence. You just have to remember the two recognition sequence. The first one is of eco-R1. Eco-R1's recognition sequence is GAATTC, and hint two recognition sequence is GTTAAC. Now, the convention for naming these enzymes was that the first letter of comes from the genus of the bacteria, and the second two letters come from the species of the prokaryotic cell or the bacteria from which they were isolated. For example, the EcoR1 comes from the this name, like E from the genus first letter and CO from the species second first and second letter, R from the rough because the rough is the strain of the bacteria, and one comes like you have isolated it first, right? Okay, from the particular bacteria in the particular laboratory. So in E-carbon, the letter R is derived from the name of the strain. The Roman numbers following the name indicate the order in which the restriction enzyme is isolated from that particular strain of the bacteria. Now, the restriction enzyme belong to a larger class of enzyme which we have already discussed. They are nucleases, and there are two types: the exo and the endonucleases. The exonuclease remove nucleotide from the ends of the DNA, while the endonuclease just make the cut at the specific position within the DNA, not from the ends, within the DNA. And as we have discussed, one more thing, that endonucleases are all of two types, है ना? First one is the type 1 and second one the type 2. Type 1 is a simple endonuclease. It is not a restriction endonuclease and it acts as methylase as well. But the type 2 is a restriction endonuclease and it does not act as methylase. We need to have one more methylase enzyme in addition to restriction endonuclease if we have the type 2 endonuclease in our cell and not type 1, right? Now, each recognition endonuclease functions by inspecting the length of the whole DNA sequence. First, you have to just inspect the whole DNA length so that you can recognize that at which particular point I have my recognition site. And once you find your specific recognition sequence, that I have just one recognition sequence in this particular length, or I have two, or I have three, then you will bind to the DNA. The enzyme will bind to the DNA at those particular point and cut each of the two strands of the double helix at specific position in the sugar phosphate backbones, right? So the each restriction endonuclease recognizes a specific palindromic sequence. So first you have to recognize, and then you have to cut the two strands and also at the specific position in this sugar phosphate backbone. I hope you know the sugar phosphate backbone. It is talking about the diester bond, phosphodiester bond or phosphoester bond. So the restriction endonuclease is breaking the phosphoester bond, or you can say phosphodiester bond. For example, you can see the sequence here is GATTC. As you guys know that this particular palindrome is of the EcoR1. So the enzyme is going to cut the both the strands at the same site. Okay, so EcoR1 cut the DNA between the bases, the G and the A on this DNA strand between G and A, and also on this DNA strand between G and A. Okay, now the foreign DNA, and this is the vector DNA. We have to cut the vector DNA and the foreign DNA by the same restriction enzyme so that they can ligate, because the same restriction enzyme is gonna produce the same sticky ends on both the vector and as well as the foreign DNA. So you must know that there should be the same palindrome on the foreign DNA and on the vector so that they can be cut by the same restriction enzyme, so that the same sticky ends are produced, so that they can easily ligate with the help of the DNA ligase. So if EcoR1 is cutting both the DNA between the G and A, so it's gonna produce the sticky end, like here only G is present because AATTC has been removed, and from in this area only AATT has sticky ends because and you can see that here only also G is present, AATTC has been removed, so it's gonna ligate over there. So it has, NCERT has shown that by the yellow arrow, that this A is gonna bind to the T. As you guys know that adenine binds to the thymine, right? So it's just gonna bind and the recombinant DNA has been produced. Please pause the video for a second and just check that this A is gonna bind to this one T, and this T is gonna bind to this A, and so like this. Okay, so the steps in the formation of recombinant DNA by the action of restriction endonuclease enzyme EcoR1. Now we are going to discuss what exactly the palindromes are. So do you know what palindromes are? There is a group of letters that form the same words when you are reading the forward direction or in the backward direction. For example, the Malayalam, against word palindrome where the same word is read in both directions, the palindrome is a sequence of base pairs that read same on the two strands when the orientation is of the reading is keep the same. For example, if you are reading from this side, so GATTC, or for example example, you are reading from this side, then also GATTC. For example, the following sequence reads the same on the 2 strand in the 5 dash into the 3 dash direction. Yes, you have to just read it in the 5 dash to 3 dash direction. So on this strand the 5 dash is this, and on this strand the 5 dash is this. This is also true if you are reading in the 3 to 5 dash direction. Yeah, for example, CTTAG or CTTAG. Restriction enzyme cut the DNA in a little away from the center of the palindromic side. Yeah, you know what? The restriction enzyme just cut the DNA because they form the sticky end. But if a restriction enzyme is cutting the DNA exactly from the center, they're going to produce the blunt end. Those are not the sticky end, right? So we have just to study the sticky end. So the restriction enzyme cut the DNA a little away from the center. For example, you can see that the E carbon was cutting between the G and A, not exactly between the A and T that was the center of the particular palindrome. So cut the DNA a little away from the center of the palindromic side, but between the same two base pair on the opposite strand. This is true for both the strand. For example, if you're going to see this was strand, then in this strand it's going to cut between the G and A. The phosphodiester bond has been broken between the guanine and adenine, and in this strand the phosphodiester bond has been broken between the guanine and adenine, right? This leaves single stranded portion at the end. This leaves the single stranded portion at the end. Single stranded portion at the end. Okay, now there are overhanging stretches. The overhanging or the single stranded portion left on the DNA are known as the sticky end on each strand. Now these are named so because they form hydrogen bond with their complementary cut counterparts. Yeah, they are gonna form the hydrogen bond with the help of the DNA ligase, so that the single stranded left out portion has the ability to become the double stranded portion. The stickiness of the ends facilitate the action of enzyme DNA ligase. Yes, if the strands are not sticky, it's not gonna like give response to the DNA ligase, so it's not gonna ligate to each other. Now the restriction endonucleases are used in genetic engineering to form the recombinant molecules of DNA. Yeah, so endonucleases are used in the genetic engineering to form recombinant molecule of DNA, because if you're not gonna cut the DNA from two different sources, how you're gonna the recombine, right? Which are composed of DNA from different source or genome. That's why it's known as the recombinant molecules of DNA. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and they can be joined together by using the DNA ligase. For example, the foreign DNA is this purple one, the vector DNA is this, and vector can be plasmid, can be bacteriophage. Yeah, there are so many type of vector. In this chapter we are going to specifically just concentrate as on the plasmid as the vector. So both the DNA are cut by the same restriction enzyme, so that they are going to produce the same type of sticky ends, and then ligase is going to join the foreign DNA to the plasmid, and you can see that the red part and slightly there is a blue or purplish part. This is the recombinant DNA molecule. And first let me tell you, the NCERT has skipped a particular thing over here, that if you're going to cut the foreign DNA, है ना, so you are just before ligating it, you have to polymerize it, right? The PCR step has been removed over here. You have to study this chapter, but they have not exactly mentioned that you just have to isolate your particular gene of interest, and then you're gonna polymerize it, right? You just can't just add it, just one strand you are isolating and just adding it. And how come you are sure that it's gonna produce a particular protein? Maybe it can't ligate, maybe it can't produce a recombinant, or maybe the ligated portion, the recombinant DNA, is not going to insert into the host, so that host is not going to become a transformant. So you just have to make the multiple copies of the particular cutted the foreign DNA, right? Now, if you are gonna form the recombinant DNA molecule, then you are gonna insert it in the host, and then the host is gonna divide. And before dividing, the host is gonna produce multiple copies of DNA, so that it's dividing, and each particular multiplied or repeated DNA is gonna divide into the babies, right? Into the child of the next generation of the bacteria. Now, you have realized that normally, unless one cuts the vector and source DNA with the same restriction enzyme, the recombinant molecule can't be created. You just have to produce the same type of sticky ends, so that they're going to ligate. If you're not going to cut them with the same restriction enzyme, you're not going to produce the same kind of sticky ends. And if you're not going to produce the same kind of sticky ends, then it's not going to ligate to each other, right? Now, the separation and isolation of DNA fragments. We know what we are going to study in this topic, that we are just going to use the agar gel, which is obtained from the red algae. And the agar gel has pores, micro pores in it, so that the DNA can cross through it, the agar gel, right? And we know that how we're gonna make the agar gel. We are just gonna add the agar into liquid, so that it's gonna settle down as a semi-solid structure, and just we are gonna create some holes in the starting point of the agar gel, so that it can produce wells in which the DNA can be kept, and then the DNA molecules are gonna travel across the pores of the agar gel based on their size. Just suppose that you are using a sieve to sieve out the dirt particles from a seed. So you know that the dirt particles are smaller in size, so it's gonna move across the sieve, the sieve pores, and the seeds, the real seeds, are gonna separated as the retentate, right? So this happens in the case of this gel electrophoresis. As you can see, the largest bands are very large in size. They can't exactly cross, or they can't move speedily through the agar gel, the pores of the agar. So the smallest is like sabse aage and the largest is sabse piche. And you know that DNA molecules are negatively charged because they have phosphate group, and phosphate PO4 3 minus has negative charge. So the negative charge always goes to the anode, because anode is named as such because anode is positively charged and anion comes on it. So this particular thing here is cathode and here is anode placed. It has not been shown in the NCERT figure. And these are the wells. These wells were created when we were settling down the agar gel, because we inserted some solid things here so that agar gel did not come in this particular area. So this space has been like created empty. We have added DNA over here in the wells, and the DNA are going to separate according to their size, according to the number of the base pair. And we already know that which DNA we have to isolate, how many base pair it contains. For example, we know that this particular column contains 200 base pair, this particular column contains 500 base pair, this particular column contains 100 base pair. And if we know that the gene of interest which we have to isolate contains 200 base pair, so we're gonna know that in which column the 200 base pair sequence has to come, and we will isolate that particular thing. And if we are gonna isolate it, then we're gonna polymerize it in the PCR machine, and then we're gonna take multiple copies of the particular DNA, and then we are gonna add it into the vector, so that the vector can recombine. So let's start now the topic, that the separation and isolation of DNA fragments. The cutting of DNA by the restriction endonuclease results in the fragments of DNA, and these fragments can be separated by a technique which is known as gel electrophoresis. Because if you're going to cut the DNA, several pieces, several fragments are being formed. So how do we know that what particular this particular fragment is my gene of interest? This particular DNA is that we can't see DNA, right? So we are just gonna isolate it through passing them through the gel of agar gel, right? Since the DNA fragments are negatively charged molecules, they can be separated by forcing them to move toward the anode under electric field through a medium or matrix. Nowadays, the most common used matrix is the agarose gel, which is a natural polymer extracted from the seaweeds. These are the red algae. The DNA fragments resolve according to the size through the sieving effect provided by the agarose gel. Hence, the smaller the fragment size, the farther it moves. Yeah, you know that the hatta katta person can't move, like can't run at a faster rate as compared to the patla dubla person. So in the case of DNA, the same thing exists, that the DNA जितना बड़ा साइज होगा DNA का, जितना उसका बेस पेयर का नंबर होगा, उतना वो पीछे रह जाएगा, उतना वो फास्टली एनोड की तरफ मूव नहीं कर पाएगा, और जितना छोटा होगा वो उतना फास्टली मूव कर पाएगा, राइट? Now, NCERT is asking that guess at which end of the gel the sample was loaded. So we know that sample was loaded in the well because the largest DNA are on this side, right? And smallest one is on this side. The separated DNA fragments. And why the name is the gel electrophoresis? Electro means some electric field has been applied. Yeah, some electric things are present here. The cathode and anode are present here, right? And it's showing that the undigested lane 1 and the digested set lane 2 to lane 4. Yes, right? So you can see that undigested fragment is standing over here. This is like थोड़ा भी digest नहीं हुआ, और ये सारे छोटे छोटे pieces है वो क्या है, थोड़े से digest हो चुके हैं, right? So हमने जो भी DNA ऊपर से डाला, हमको क्या पता कि हमने उसको जो कट किया वो कट हुआ या नहीं हुआ, right? तो जो सबसे बड़ा आपको यहाँ पे दिख रहा है, ये कट बिल्कुल भी नहीं हुआ था, सबसे पीछे है, और ये सारे इससे ज्यादा छोटे लग रहे हैं, कट हो चुके थे, और अब इनका भी जो कटेड पार्ट था उसमें से वो सारा पार्ट अलग अलग एक दूसरे के साइज के अकॉर्डिंग, एक दूसरे के बेस पेयर के अकॉर्डिंग अलग अलग हो रहा है. The separated DNA fragments can be visualized only after staining the DNA with a compound which is known as ethidium bromide. You can't just see the DNA with your naked eye. You have to add some pigment, ethidium bromide. And you can't see the DNA even after adding ethidium bromide if you are watching it in the visible light. So you have to add the UV ray radiation. You can't see the pure DNA in the visible light without staining. Now you can see the bright orange colored bands of DNA. DNA looks orange colored in the case of if you are going to pass the UV radiation in a ethidium bromide stain gel exposed to UV light, right? Okay, and the separated bands of DNA are now cut out from the agarose gel. You gotta cut out and extract from the gel piece. This step is known as elution. What is elution? The cutting out of the DNA fragment from the agar gel, right? And the DNA fragments purified in this way are used in constructing a recombinant DNA molecule by joining them with cloning vector. Okay, I hope this topic is clear. we will see the next topic in the next video, that is the cloning vector. And we're going to discuss the PBR322 in this, and then we're going to discuss the recombinant host, and yeah, then we are going to discuss that how we are going to isolate and cut, and yeah. Okay, so thank you so much for watching this video. If you guys uh, came to understood the topic from the NCERT, then make sure to like and subscribe to my channel.